Single RNA polymerase in Escherichia coli. In bacterial systems like E. coli, a single RNA polymerase, in short RNAP, species is responsible for synthesis of all kinds of RNAs, mRNA, tRNAs and rRNA. This RNA polymerase has been purified and its structure and function is now known in some detail. The E. coli RNA polymerase has a molecular weight of about 4,80,000 Daltons and consists of 5 polypeptides including 2 chains of alpha polypeptide, 1 chain each of beta, beta prime and sigma polypeptides, the details for which are given in table as well as in the figure. The beta prime subunit is the largest subunit encoded by RPO-C gene. It contains part of the active center that is responsible for RNA synthesis and also contains some of the determinants responsible for non-sequence specific interactions with the DNA nascent and RNA. The beta subunit is the second largest subunit encoded by RPO-B gene and contains the rest of the active center that is responsible for RNA synthesis. It also contains the rest of the factors for non-sequence specific interactions with the nascent RNA and DNA. The third largest subunit is alpha subunit and is present in 2 copies per RNA polymerase. Each alpha subunit contains 2 domains, alpha NTD that is N-terminal domain and alpha CTD that is C-terminal domain. The alpha NTD contains the determinants for assembly of RNA polymerase and alpha CTD contains the factors for interaction with the promoter DNA, making sequence specific interactions at upstream element containing promoters and non-specific non-sequence interactions at promoters. This is clearly seen in this figure. It also contains determinants for interactions with the regulatory factors. 2 of these are identical. Thus, the enzyme contains 4 distinct polypeptides. The RNA polymerase molecule, the holo enzyme, has the composition alpha 2, beta, beta prime, sigma, in which attachment of sigma factor is not very resistant, so that the core enzyme consisting of alpha 2, beta, beta prime can be easily isolated. The alpha subunits are involved in the assembly of the tetramer core of RNA polymerase. The beta subunits contains the ribonucleoside phosphate binding site and the beta prime subunit harbors the DNA template binding region. One subunit, the sigma factor, is involved only in the initiation of transcription. It plays no role in elongation. After RNA chain initiation has occurred, the sigma factor is released and chain elongation is catalyzed by the core enzyme consisting of alpha 2, beta, beta prime. The function of sigma is to recognize and bind RNA polymerase to the transcription initiation or promoter sites in DNA. The core enzyme, where sigma is absent, will catalyze RNA synthesis from DNA templates in vitro. In doing this, it will initiate RNA chains at random sites on both strands of DNA. In contrast, the holo enzyme, where sigma is present, initiates RNA chains in vitro only at sites used in vivo. The sigma factor helps in the recognition of start signals on DNA molecule and directs RNA polymerase for the selection of the initiation sites. In the absence of sigma, core enzyme initiates RNA synthesis in a random manner, suggesting the role of sigma in recognition of initiation sites. Once RNA synthesis is initiated, sigma dissociates after RNA is 8 to 9 base pair long, and then the core enzyme brings about the elongation of mRNA. The dissociated sigma factor may again combine with the core enzyme to form RNA polymerase holo enzyme, where sigma is present. The alpha and beta prime have constant sizes in most bacteria. The sigma varies from 32,000 to 92,000 Daltons. Although in prokaryotes like E. coli, all RNA synthesis is done by only one kind of RNA polymerase molecules, there may be more than one sigma factors which associate each with the same core enzyme at different times for expression of different genes. For example, in E. coli, besides sigma 70 used under normal conditions of growth, at least 3 other sigma factors, that is sigma 32, sigma 54, sigma 28 are now known, which are used under adverse conditions like high temperature, nitrogen deficiency and for chemotaxis. The core enzyme contains the catalytic site. This catalytic site contains 2 metal ions, that is magnesium. In its active form, one metal ion remains bound to the enzyme, whereas the other metal ion appears to come in with the nucleoside. 3 conserved aspartate residues of the core enzyme participate in binding these metal ions in catalytic site. The overall structure of RNA polymerase and DNA polymerase are quite different. Their similar active sites are the products of convergent evolution. Sigma subunits of RNA polymerase recognize promoter sites. The tetrameric core consisting of alpha 2 beta beta prime of RNA polymerase is unable to start initiation of transcription at promoter sites without sigma factor. Rather, the complete holoenzyme consisting of alpha 2 beta beta prime sigma is essential for initiation of transcription at the correct start site. The sigma subunit contributes to specific initiation of transcription in 2 ways. First, it decreases the affinity of RNA polymerase enzyme for general regions of DNA by a factor of 10 to the power of 4. The core enzyme binds the DNA indiscriminately and tightly in its absence. Secondly, the sigma subunit enables RNA polymerase to recognize the promoter sites. A large fragment of a sigma subunit was found to have an sigma helix on its surface. This sigma helix has been implicated in recognizing the 5' TA TAA T sequence of the -10 region. The holoenzyme binds to DNA duplex and moves along the DNA double helix in search of a promoter, forming transient hydrogen bonds with exposed acceptor groups and hydrogen donor on the base pairs. This process is rapid because RNA polymerase slides along DNA duplex instead of binding repeatedly and dissociating from it, or rather than in 3 dimensions. The promoter site is encountered by a random walk in 1 dimension. The constant rate observed for the binding of RNA polymerase holoenzyme to promoter sequences is more than 100 times as fast as could be accomplished by repeated encounters moving on and off the DNA. The sigma subunit is released when the nascent RNA chain reaches 9 or 10 nucleotides in length. After its release, it can assist transcription initiation by another core enzyme. Thus, the alpha subunit acts catalytically. E. coli contains multiple sigma factors to recognize different types of promoter sequences contained in E. coli DNA. The sigma factor that recognizes the consensus sequences is called sigma 70 because it has a mass of 70 kilodalton. When the temperature is raised abruptly, a different sigma factor comes into action in initiation of transcription. E. coli responds by synthesizing sigma 32, which recognizes the promoter sequence of heat shock genes. These types of promoters exhibit -10 sequences that are somewhat different from the -10 sequence for standard promoters. In the following figure, a comparison of the consensus sequence of standard promoters sigma 70, heat shock promoters sigma 32 and nitrogen starvation promoters sigma 54 of E. coli is shown. The increased transcription of heat shock genes leads to the synthesis of series of protective proteins. Other sigma factors respond to extreme environmental conditions such as nitrogen starvation. These findings demonstrate that sigma factor plays a key role in determining where RNA polymerase enzyme initiates transcription.